Hello everyone, and welcome back for more biotechnology. In this video we're going to specifically talk about the use of antibodies in medical technologies. So we've already covered in previous chapters and previous videos some very important, very useful biotechnological applications that take advantage of the affinity that antibodies have for particular proteins. So we talked about ELISA assays, we talked about Western blot assays. Basically, the idea that proteins bind to particular antigens with very high affinity makes it very easy to detect the presence of particular molecules or particular proteins in a given solution or a given mixture of different compounds. So antibodies are also called immunoglobulin. Your textbook says that they're Y-shaped, so this is only in the case of a particular type of immunoglobulin called IgG. If you look up any of the other immunoglobulins, you'll see that they're not necessarily Y-shaped. But they consist of 4 different polypeptide chains, 2 of which are called light chains and 2 of which are called heavy chains. So as we know by now, what makes antibodies so useful is that they bind to antigens. Usually those antigens will be some type of protein with very, very, very high affinity. And physiologically speaking, with regard to the immune system, when an antibody binds to a particular antigen, usually as part of some foreign invading pathogen, the antibodies will recruit the immune system, the cells of the immune system, the white blood cells, where this interaction occurs in the body so that the infection or the pathogen can be fought and destroyed where it stands. And as I mentioned before, we've already seen how useful antibodies can be in vitro applications, where we've already seen the usefulness of things like ELISA and Western blotting. So let's introduce a new biotechnology application for antibodies. This is called flow cytometry. Now, it is important to clarify that flow cytometry itself doesn't necessarily require the use of an antibody, but antibodies actually can be very useful here. So if we decide to use antibodies for flow cytometry, we can take a suspension of cells and introduce a particular antibody that recognizes a protein that is on the surface of some cells but not most cells. Now, usually speaking, this antibody that is bound to the outer part of the cell will be labeled with a fluorescent dye, and then as the cells pass through a narrow opening in the flow cytometer will pass a laser that should excite the fluorophore on that particular antibody, and everything is nice and programmed and well set up so that the cells that fluoresce, meaning the cells that are labeled with antibodies, get sorted in one direction, and all the other cells that do not fluoresce get sorted into another. The unlabeled cells do not react. So this type of application of flow cytometry, in which we can sort cells based on their characteristics like whether they have an antibody bound or not, is called fluorescent activated cell sorting, or FACS. And like I said, this allows us to separate and collect cells of different types depending on which cells are labeled and which ones are not. Now, as I said, flow cytometry itself does not necessarily require antibodies. The lasers that pass through the sample of cells that pass through the opening here can be assessed on the basis of how they reflect and deflect light. And we can assess things like how healthy the cells are, how big they are they alive, are they dead, how much granularity do they have inside the cell. So you don't necessarily need antibodies here, but antibodies can be made use of quite well. So let's talk a little bit about how antibodies get manufactured in biotechnology terms. The old-fashioned way of doing this, if you decide that you're going to make an antibody against a particular antigen, usually a protein, so let's say that you're studying a particular protein and you need a way of detecting that particular protein, right? So what if there is no commercial antibody available that was raised against that protein? Well, if there's no antibody to bind to that protein, you've got no way of detecting the protein. So what do? Well, you make the antibody yourself. So what you're going to do here is you are going to create a peptide. We talked about that in the previous video. You're going to synthesize a peptide that corresponds to the part of the protein that you're interested in that you want your antibody to react to. And you are going to inject that peptide into an animal. The most common animals that are used for making antibodies are mice, rats, rabbits, and donkeys. Goats you could also throw into that mix as well. So when you inject an animal with that particular antigen, that animal and their immune system is going to start generating antibodies against the invading pathogen, or what they think the invading pathogen is. In reality, it's just the peptide that we injected into their bloodstream. After we have given the animal sufficient time to mount an immune response, their blood should now contain a very high concentration of antibodies against the peptide that we injected. We then exsanguinate the animal, meaning we drain them of their blood. Unfortunately, that's fatal, so I'm sorry if you're not happy to hear that. But we collect their blood, and then we can purify the antibody from their blood, and we purify the antibody by affinity chromatography. We essentially conjugate the peptide that we synthesize to a resin, we pass the blood through the column, and we collect the antibodies. You'll note in this diagram that one particular type of immune cell is responsible for making antibodies, and these are called B lymphocytes, or sometimes just called B cells. The way I described the antibody production process is how you make what are called polyclonal antibodies. If you have a polyclonal antibody, it means that not every antibody in the mixture of antibodies is exactly the same. Some of them will bind to the antigen with very high affinity, some of them may not, and some of them may even have off-target effects. But usually this isn't that big of a problem. Now, you can also make monoclonal antibodies, which are vastly superior in not only their affinity, but also their specificity for the antigen that they're supposed to bind, but making them is much, much, much more difficult. You have to ensure that all of the antibody production is being done by one specific type of B cell, and you want every B cell that is making antibodies to be absolutely identical to all the other B cells. So the actual protocol for doing this is complicated, and we're not going to get into it in this context. So let's talk about an obvious application of antibodies, and that is vaccines. Now, since we're in the middle of the COVID pandemic, as I record this video on November 19th, 2020, we obviously are dying for a COVID-19 vaccine, right? And it sounds like one is right around the corner. So to educate ourselves just a little bit, let's talk about how you actually go about making a vaccine. Now, most vaccines, not all of them, but most vaccines are going to seek to generate an antibody response in a very particular way, and that is essentially the same way that we did it with animals. We are going to knowingly expose the host organism to an antigen from whatever we're trying to mount an immune response against. In the case of COVID-19, we would want our antigen to be a protein from the actual coronavirus that causes COVID-19, the SARS-2 coronavirus. So oftentimes the antigen will consist of a weakened or inert virus or some bacterial component. Like we said, that's usually going to be some protein, right? Some protein that is specific to that particular organism or virus. So the idea here is that after vaccination, the body's B cells should begin generating antibodies against that particular antigen, eventually mass producing those that bind to the antigen of interest. The initial antibody response may not necessarily specifically target the antigen, but eventually the body will wise up and will figure out which antibodies work the best, and they will start manufacturing just those antibodies. So antibody production is eventually going to die down, so once the antigen has been gone for a while, the body's not going to feel the need to have that particular antibody around for forever. So the question is, okay, what good does that do? Well, thankfully, your body has what is called a memory response, meaning that essentially your immune system is going to remember what it looks like when that antigen comes in again, such as if you get infected again, and your body is going to know, okay, I've seen this antigen before and I know exactly how to deal with this sucker. I'm going to produce this type of antibody right here, and that antigen is not going to stand a chance. So your B cells are going to remember what that antigen looks like, and they'll know exactly what kind of antibody to make to counter it right away. So here's another diagram from your book that illustrates what the vaccine process is like. So whoever is going to be vaccinated will be injected with a particular antigen. The B cells will recognize the foreign antigen and begin making antibodies against it, and the antibody response will recruit the immune system such that we can destroy the foreign invading cells and break down those antigens. So everything that we talked about is illustrated here in a fairly simple way. Now, a common question that I get, regardless of what class I'm teaching, whether it's just general biology or human physiology, or I would expect to get it in this class as well, if we're talking about vaccines for something like influenza, why do you have to get a new vaccine every year? Now, some vaccines generate immunity that lasts a lifetime, right? So, for example, the polio vaccine. You only have to get that once when you're a little bitty child, right? You never have to get it again. Some vaccines you have to get like once every 5 or 10 years, like your tetanus shot, right? And then some vaccines you have to literally get every single year. So why is that? Well, there's a couple of explanations. Now, in the case of the tetanus shot, every subsequent tetanus shot is what's called a booster shot. It's meant to just stimulate the immune system and remind the B cells of what the antigen looks like, so basically they don't forget. Now, if we're talking about why you have to get vaccinated with a flu shot every year, that's not really what we're talking about here. So the influenza virus basically will become immune to the previous year's vaccine through genetic mutations. Now, we didn't get to talk a whole lot about viral genetics earlier in the semester, but viruses do contain a genetic code in the form of DNA or RNA, and as always, that DNA or RNA codes for particular proteins. We've also talked about genetic mutations and how if you change the coding sequence of a piece of DNA or RNA, you're going to change which amino acids get incorporated into the protein. So, if the previous year's influenza vaccine uses an antigen from the previous year's virus, well, next year's virus may have mutated so that that antigen is no longer applicable. It's basically obsolete at that point. So, a lot of what scientists who study the influenza virus are doing all year round is they are trying to predict what kind of mutations the flu virus is going to make, so that we can make new vaccinations that will counter it, so that we greatly reduce the number of people that are going to get sick. So as we said, the virus does contain a number of proteins that are potential vaccine targets. We can inject those into someone and get them to mount an antibody response. But like I said, if the virus mutates and changes these proteins, that antibody from last year may not bind anymore. So if the virus is going to be sneaky and try to mutate to circumvent last year's vaccine, we need to be just as sneaky to try to stay one step ahead of the virus. All right, so that is going to do it for this video on antibodies and vaccines and other sorts of applications that involve antibodies. So I enjoyed it. I look forward to seeing you for one more video in Chapter 12. I will see you next time.